Also nice to see so many of his former and present co-workers participate in this symposium. So without going any further into that, I will invite the first speaker, Kumar Sundaram of our microbiology cell biology, to give his talk on cancer stem-like cells, understanding tumor cellular hierarchy and heterogeneity. Professor Kumar Sundaram.
Thank you. Thank you,
Sundaram,
Chair.
can you start?
Yes, I will. Thank you, Chair, for the kind introduction. Let me also start thanking the IPC faculty members and the Chair for inviting me to this wonderful meeting, and Professor Ashok Samuelson have been known to me for last 22 years. That duration of my presence in the institute. From the very first year I joined in MCB 99, we have been working together on various aspects related to chemistry, and I do have quite a few slides at the end of it to show some of the good papers which came out of these collaborations, and I will say a few words about how those collaborations went and what kind of the way by which we did the interactions, and I'll try to give you some detail about that. But let me start with my actual talk today. The title of the talk, as you can see, Cancer Stem-like Cells, Understanding Tumor Cellular Hierarchy and Heterogeneity. Let me take a couple of minutes to introduce the title to all of you. A few seconds, please. Okay, so you all know the cancer cell lines. I'm sure many of the chemistry faculties and students would know, those who are working on chemistry relevant to biology. Cancer cell lines have been used for many decades as a model to describe cancer. They are not bad. Actually, cancer cell lines are very good because they have the ability to grow continuously, perpetually. They never stop growing as long as we provide nutrients, and they can also make tumors in animal model. So they have really have useful in studying cancer and trying to develop therapeutic molecules, etc. But very often the successes have never been translated to human patients. When they try to apply to human patients, many of those discoveries have failed. Okay, there are many reasons why they have failed. One of the reasons I want to show you here, if you look at this, is a cross section of a brain, and the cellular area, what you see here in the darkly stained cells, which is encircled here, which shows the presence of a tumor, a brain tumor. Okay, if you look at this particular section under the microscope, the tumor is made up of tumor cells, but they are not just made up of only tumor cells. Okay, there are many other cell types are there. But in the cell line model, we are only using the tumor cells only for our study. But the tumor is not made up of only tumor cells. So what are they? What is it then made up of? So if you look at a schematic of how a tumor look like, what you see in the middle is actually tumor cells. There itself, there are two types of tumor cells. What you see are the red color cells are the cancer stem cells. You can call them as a cancer stem-like cells as well, and also cancer stem cells. And there are a large number of blue color cells. They are also tumor cells. They are called non-cancer stem cells, otherwise called differentiated bulk cells. So there is already two types of tumor cells. In addition to that, there are stromal cells. These are cells which are present in and around the tumor. Okay, you can see there are immune cells which comprises of lymphocytes of different types, macrophages, and there is also blood vasculature which consists of endothelial cells, pericytes, and there are also normal cells of the particular organ. For example, if it is a brain, normal astrocytes are there. So there are many number of type of cells are there in the tumor. The tumor growth is ultimately controlled by the interaction between all the type of cells which are present in the tumor. So anything you develop targeting only the tumor cells, that's the reason why it doesn't seem to work in the actual clinical trials. So now it's a time for me to introduce the title. I said cancer stem-like cells. I will tell you what it is. And then there is also two terminologies: tumor cellular hierarchy and heterogeneity. Hierarchy and heterogeneity. Cellular hierarchy and cellular heterogeneity. What do I mean by that? Okay. Now, if you look at hierarchy, I said in the previous slide, in the tumor there are different types of cells are there. So the stem cells, the non-cancer stem cells, and so many stromal cells. Which of them is more important? All are important for the tumor. Which one is more important than the other? How do you answer that? If you take a tumor, separate all these different type of cells in the tumor, and then ask which type of cell can actually initiate a tumor if you give a chance to begin with. Okay. It is one and the only cancer stem cells. Cancer stem cells alone can initiate a tumor. If you separate out different type of cells from a tumor and individually inject different type of cells, only cancer stem cell can initiate a tumor. No other cells can initiate a tumor by themselves. That is why people call cancer stem cells are at the top of the hierarchy, because they alone can initiate a tumor. So that is what I meant by cellular hierarchy, wherein cancer stem cells sit on the top of it. Number two, heterogeneity, cellular heterogeneity. As I said in the previous slide, there are reasons why there is a cellular heterogeneity. One is, to begin with, there are different types of cells are there. So heterogeneity exists in terms of different type of cells. Second is, there is a new phenomena which people have identified called tumor cell plasticity. That is, one type of cell can become another type of cells. For example, CSCs, cancer stem cell, can actually differentiate to form non-cancer stem cells. And then it can also de-differentiate to form stem cells. One more phenomena people have recently found, non-cancer stem cells can actually differentiate further to form endothelial cells. So these two phenomena actually changes the proportion of different types of cells in the tumor, which can vary between the different tumors, that carries a heterogeneity. So what I mean by hierarchy, cellular hierarchy and cellular heterogeneity, I guess I have introduced to all the audience. And the overall title is actually cancer stem cells, because they are the one which can initiate a tumor. The whole world is going behind trying to purify and study, understand the biology behind cancer stem cells. Okay, so cancer stem-like cells or cancer stem cells alone can initiate a tumor. They are called by different names: cancer stem-like cells, tumor initiating cells, tumor propagating cells. Different names are used by different laboratories. The question I want to put forth in front of the audience is coming to the next slide. That is, if you look at a tumor, I hear some background voice. Hello.
No problem. Continue something. Sundaram, your mic is on mute.
Okay, you have not been listening to me so far?
No, we have been listening. Go
Okay,
on.
I'm sorry, it has happened by mistake. Okay, so if you look at this particular structure as a tumor, and if you try to identify how many stem cells or non-cancer stem cells are there, cancer stem cells, although they are the most important cells that can initiate a tumor, they only form a tiny proportion of the tumor. Bulk of the tumor are made up of green cells, which are non-CSCs. About 99% of the tumor is made up of non-cancer stem cells. Okay, and now if you look at it, how is it possible when the stem cell can alone form a tumor? How at the end of a tumor is being formed, majority of stem cells tumor is made up of non-cancer stem cell. How does it happen? How exactly the tumor is evolved to make huge bulk amount of the non-stem cells? So if you look at in the next slide, so let's look at the tumor initiation and the growth. Let's say there is one cancer stem is formed that divides into two becomes 4. Then simultaneously it also differentiates to form the bulk cells, the non-cancer stem cells. So this process continue to happen. The differentiation is the direction towards differentiation is more often happening. At the end, you will have the whole tumor made up of majority of the non-cancer stem cell, only a tiny proportion of the cancer stem cell, which is sufficient for cancer stem cell, because cancer stem cells are known to be resistant for most of the therapy and most of the very difficult conditions, like not having enough amount of nutrients, etc. So they can remain alive. Even though non-stem cells can die, they can remain alive and continue to sustain the growth of the tumor. That's how the tumor continues to remain there in the tumor. So now the question I said in the previous slide is, so CSCs alone can initiate a tumor. It's very clear. And because if you purify different cells and inject individually, only the CSCs can initiate a tumor. Non-CSCs cannot initiate a tumor. If you put 100 times more non-CSCs, you cannot form a tumor. But the question is, non-CSCs form the most of the tumor, 99% of the tumor. And non-CSCs, the question is, are they there to form the bulk? If they can't initiate a tumor, are they there to form the bulk? Or do they have any function? This is a question we asked. Okay. So in our lab, we work on glioma. It's one of the common brain tumors. So the stem cells coming from the glioma is the glioma stem-like cells. They are called GSCs. I will not use this terminology anymore. I'm just introducing to you because this is what we actually work on. So now, in order to understand CSCs and non-CSCs, how do they function, the relevance of non-CSCs, what we did is, we looked at secreted proteins from CSCs and non-CSCs and analyzed their composition by proteomics. We used a label-free quantitative proteomics using nano LC-FT MS. We identified several proteins which are present at high abundance both in CSC secretome and non-CSC secretome. Our interest is primarily on looking at proteins which are in high abundance in the CSC secretome. These are proteins which are secreted at high levels by CSCs. Many projects are going on in the lab. However, one protein which got our attention, which is called FMOD, fibromodulin, which is present at high abundance in the non-CSC secretome. Okay, now we asked a question, why is this protein is secreted by non-CSCs to high levels and not expressed at all by CSCs? Why we got interested into this protein? We have already worked on this protein in a different context, published a paper in Oncogene. We showed this particular protein is expressed by so-called established glioma cell lines, large amounts, and that particular protein is able to activate glioma, induce the glioma cell migration, although that does not affect the cell proliferation. So this particular protein is able to induce migration, but does not affect proliferation. This is all the only information we have. Now our new findings show this particular protein is expressed only by the non-cancer stem cells and not by the stem cells. Okay, so it is our interest to find out what is the function of the fibromodulin, which is secreted only by the non-cancer stem cells. Okay, what is the role of non-cancer stem cells secreted FMOD in tumor growth? Okay, I'm only going to show you two experiments which are carried out using animal model in detail, and no other experiment I'll be explaining to you. Just make it understand the finding, the function for this non-CSC secreted FMOD in tumor growth. Okay, now we made use of a particular experimental protocol used by this paper. In this paper, what they show here is there is a reciprocal signaling between glioblastoma stem cells and the non-cancer stem cell. If you consider red cell is a CSC and blue cell is a non-CSC, they demonstrated non-CSCs can also secrete certain factors which can go on to help activate the cancer stem cell in its function of maintaining the tumor growth. So they already proven non-cancer stem cells can actually help the stem cells. So the experiment, what they did is they did a co-implantation experiment. What they did is they took some mouse intracranially onto the brain. They injected CSCs and CSCs and some amount of non-CSCs, and together they looked at the growth of the tumor. These cells are actually tagged with luciferase, so one can measure luminescence, the light emission, as a measure of the tumor growth. When you inject only CSCs, you can see hardly any tumor being formed at this particular time frame of the experiment. You can see a small tumor is formed here, a representative animal section. Whereas when the stem cells were co-injected with the non-cancer stem cells, you can see the same number of 10 power 3 CSCs can form a much bigger tumor. This does not happen if you co-inject fibroblast. And if you inject only non-cancer stem cells, again, there are no tumors. So what it tells you is the non-cancer stem cells can actually help the growth, promoting the growth of the tumors which are initiated by the stem cells. Okay. Now we asked a question, when you do this experiment, you take this non-cancer stem cells, silence FMOD, that is using RNA interference technique, you can specifically inhibit the expression of FMOD in the non-cancer stem cells and then inject those cells along with the cancer stem cells, see whether non-cancer stem cell will continue to help the cancer stem cells in promoting making the tumor or not. That is a question we asked. So a simple experiment, what we did is we took a particular mouse glioma cell line, which is having a luciferase tagged version, in which we introduce doxycycline inducible version of RNA interference construct for FMOD, and also it has a M-cherry. So whenever you add doxycycline, the particular cell line will express M-cherry, which will be you can use fluorescence scanning to measure the tumor location and the size, and the silencing also will be induced. That means FMOD will be silenced. The protocol is like that. You take bunch of mice, inject CSCs along with the non-CSCs which are having non-targeting shRNA. Number two, CSCs along with the non-CSCs with the FMOD silencing shRNA. Okay. So of course we have a control CSC alone and non-CSC alone. So day 0 we inject, right? And then after day 9, we continue to image for M-cherry and bioluminescence. And we also at 9th day, we divide them into 2 groups, doxycycline, no doxycycline, and then continue to see whether in this particular case, in the plus doxycycline, FMOD silencing will happen, whether it will affect the the help given by the non-CSE to CSEs. You can see here complicated experiment. You can see the SHNT, SHF mod combination experiment at up to 9 days when doxycycline is not added. You can see tumor is formed very nicely in all the 4 groups. And then the next one, you can see the growth and then bioluminescence. And then each group is divided into 2 groups, minus dox, plus dox, again minus and plus. You can see here, in the left side SHNT, you can see whether dox is added or not. Bioluminescence shows that tumor continue to grow. And also on the right side, you can see the M cherry is coming up in the tumor. Whereas on the right side, where SHF mod construct is there, even plus doxycycline is added. Tumor growth initially is there, M cherry is also there, but subsequently it comes down, but not in the minus dox. Of course, these controls do not form a big tumor, and the quantitation is shown here. Basically, you can see here, CSCs alone form a tumor very slowly, but particularly when F mod is silenced by adding doxycycline, non-CSCs are not able to collaborate with or help the CSC generated tumor to grow further. That is a red line, which starts off initially and then comes down as low as only non-GSCs. So basically, F mod silence non-CSCs fail to support tumors initiated by CSCs. So some unknown functions maybe for this F mod. We went on to show that it can induce angiogenesis. We also looked at signaling pathway happening in the endothelial cells. We identified there are 2 pathways, the Notch pathway and the integrin pathway is very important. I'll skip that portion. One last experiment, the co-implantation experiment, you are actually mixing 2 different types of cells and co-implanting into the tumor, in the animal. But non-GSCs are not formed like that. They are formed de novo. When the CSCs are forming the tumor, they are formed through differentiation. So this is how it happens. So stem cells get then mutated to non-stem cancer stem cells, and then it divides and differentiates, and the non-stem cells are formed de novo by differentiation. So suppose if you can silence F mod in the non-CSCs as they are being formed, okay, will it affect the growth of the tumor? That is a experiment we did next. Instead of co-implantation, again we resorted to the animal model. We took CSC cells now here. In that same construct is introduced where you have doxycycline inducible M cherry and shRNA construct for F mod. And then these were introduced, and about 20 to 19 days, the tumors are allowed to grow. These 2 groups divided into 2 groups. Now 1 minus and plus dox. You can see here, this is immediately after doxycycline injection. Before doxycycline will induce RNA interference. You can see both SHNT and SHF mod, both of them have these similar amount of tumor growth in the brain. And then subsequent scanning, you can see in the SHF mod condition, doxycycline would have induced SHF mod construct, tumor started decreasing, while on the next XHNT, the tumor continued to grow. You can see the quantitation here. When you silence F mod, the tumor doesn't grow any further, which means de novo formed non-CSCs, if you silence for F mod, they no more help the growth of the tumor, which are initiated by CSCs. And you can see that tumor presents large amounts in the control animals, not in F mod silence condition. And again, the growth of the survival of the animal increases if you silence F mod as compared to the control animals. So basically, we also show that angiogenesis is the one which is compromised when you actually silence F mod compared to the control animal that is shown here. So now I come to the model what we propose. In this, basically, if you consider this is a glioma tumor, it's also called glioblastoma. You have 2 types of cells, cancer stem cells and then the non-cancer stem cells. Non-cancer stem cells form the bulk of the tumor. And non-cancer stem cells make this protein F mod. The F mod actually interacts with the endothelial cells through particular pathway we have defined, and it is able to activate the angiogenesis. Thereby, it is creating a highly vascular glioma tumor, thereby helping the overall growth of the tumor and being more aggressive. This is our model. And the take-home message or the overall conclusion is, in the tumor, there are not only tumor cells. The tumor cells also, there are 2 types. Everybody has appreciated cancer stem cells and non-cancer stem cell. Whole world is looking at cancer stem cell. What we are saying, yes, we need to look at target the cancer stem cell, but let us not ignore the so-called non-cancer stem cells. Targeting should continue to be done for the non-cancer stem cells as well, because our study clearly demonstrates non-cancer stem cells also contribute to the overall tumor growth, even though they cannot initiate tumor. In addition, other stromal cells also has to be targeted. Now, coming back to the last few minutes, chemical biology collaborations with the Professor Ashok Samuelson. We are here to felicitate Professor Ashok Samuelson for his 3+ more decades of his fantastic scientific work in our institute. And the last 22 years, I've been working with him for various projects related to metals like ruthenium, copper, gold, and few other. And these are the bunch of students involved in this collaborative project. Those who are colored red are graduate students from Professor Ashok Samuelson's lab, who actually helped to do this work. Those who are colored black are students are postdocs who worked in my lab, who actually participated in this project. So I just wanted to show you a few slides where some of these important papers came out through this fruitful collaboration. One of the earliest published through this collaboration was related to copper complex showing anti-cancer activity. Nasrat was the graduate student from Ashok's lab, and Purniya Padak was a postdoc from my lab. We showed very nicely looked at the mechanism behind the cytotoxicity by this compound copper complex, and we show very nice pictures that they are able, the copper complex is able to make DNA damage, induce apoptosis, and also it is able to induce p53 and its target gene p21, and able to inhibit DNA synthesis. Nice cell cycle analysis show that copper complex induces not only cell cycle arrest in G1 and G2, and also induce a huge amount of apoptosis. And this is one of the very highly cited papers. And Journal of Inorganic Biochemistry, another paper where it is based on ruthenium complexes. Sangeeta Das, another graduate student from Ashok's lab, and Sarika Sinha was a postdoc in my lab. They demonstrate, using BRD incorporation, DNA synthesis which is happening normally in the control cells gets decreased when you treat them with the ruthenium complex which used in this particular study. It is also able to induce invasion by the cancer cells. And the next paper, Journal of Medicinal Chemistry again, this is again coming back to copper complexes. Turai Pandi, another graduate student from Ashok's lab, and we basically showed both in vitro, in vivo using Newton mice model, we are able to demonstrate its anti-cancer activity of this copper complex. And next paper, Journal of Biological and Inorganic Chemistry, Disha Dayal and Turai Pandi, and there were actually two papers which I'm showing. They interchange their position in terms of first authorship, and basically we demonstrate ability of using intracellular zinc for fluorescent imaging of cells. Okay, this is these two papers are also very highly cited paper. Turai Pandi was a graduate student from Ashok's lab, and Disha Dayal was a postdoc from my lab. And yet another paper where Raja Mitra was a first author, and Sangeeta Das also participated in this work. Here we looked at ruthenium complex and its ability to inhibit cancer cells. And they basically demonstrate ruthenium complex is able to inhibit topoisomerase 2 activity. If you can see here, this is actually the control plasmid DNA is supercoiled, and if you add topoisomerase 2, it is able to relax. The formation of multiple bands tells you that topoisomerase is able to relax the supercoiled DNA, and that can be inhibited by etoposide, which is a known topoisomerase 2 inhibitor. You can see here different forms of ruthenium complex are also to varying extent is able to inhibit the topoisomerase 2 activity. And that is actually the gist of this particular paper. By virtue of its ability to inhibit topoisomerase, we found that it is actually inhibiting cancer cell growth. And the last paper I would like to share is that where the first author is Anup. Anup was a graduate student from Ashok's lab, and Siddiq Zuman is actually the postdoc, worked in my lab. They worked together. And gold nanoparticles, biotin decorated gold nanoparticle carrying copper complex was used both in vitro and in vivo study to demonstrate their potential to inhibit the growth of cancer cells both in vivo and in vitro and in vivo using Newton mice model. So these are some of the papers I wanted to share with you. And overall acknowledgements, of course, I have quite a lot of students in my lab and collaborators from biology work, and certainly Professor Samuelson's have a place there, and all his wonderful students, Nasrat and Sangeeta, Turai Pandi, Raja, Anup. I always try to make a point to mention our students, because any scientist accomplishments would not be possible without their good students, their hard work and papers published. And so I put the name here, Nasrat, again to repeat, Nasrat, Sangeeta, Turai, and Raja and Anup, and perhaps I would have missed out some of the names, but I will stop here, and thank you for this opportunity. I will come back again during felicitation to say a few words about Professor Ashok. Thank you. If there are any questions, I'll be happy to take.
Oh, thank you, Professor Sundaram. The talk is now about brief. One or two questions, please.
Yes,
I think
please.
Nusrat has a question.
Yes.
Thank you, Professor Kumar,
Yes.
for your wonderful talk. I'm just curious to know, when we are using the normal anti-cancer drugs, are they able to cross the membrane of cancer stem cells? And if not, do you think there is a difference in the membrane structure of cancer stem cells or something else?
Yes,
Or
I
do
don't
you
know
have
about,
any comments?
yeah, I don't know about membrane structure as it is. I'm not qualified to answer that. But all I know is there are certain transporter proteins which are there in the membrane, which are kind of, their composition is little different in the cancer stem cell compared to the non-cancer stem cells. They are known to take them out. Okay. The drugs, and in fact efflux them out. In fact, that property is used, one of the ways to identify the cancer stem cells. So if you actually do sorting, you take the entire population of cells from the freshly resected tumors, add these drugs which can be efflux them out, and there is something called site population which comes at one particular location in the flow sorter. These set of cells have the ability to push them out, efflux them out more efficiently than the non-stem cells, and thereby one can identify cancer stem cells. That is one of the mechanisms, reasons why cancer stem cells are more resistant to majority of the many known drugs. So this is what I can answer, whatever I know, Nasrat.
Thank you so much.
Are there any more questions? So, please join me in thanking Professor Sundaram for his well.
Thank you.
I requested Professor Sivashankar from Pondicherry University to give his talk on the Sivashankar on the copper in nitrogen and carbon chemistry, catalyst or co-catalyst. Professor Sivashankar. Professor Sivashankar?
Yes, sir. Yes, sir.
You can put in slideshow mode and start.
Yes, sir.
Yeah.
Now?
Yeah, you can go ahead.
Okay, sir. Thank you, sir, for your kind introduction. And also, I would like to thank the IPC department organizers for conducting this one-day symposium. Truly, I am honored by giving this talk on this occasion of my Professor Ashoka G. Samuelson on this particular occasion. Okay. Today I will be talking to you about copper in nitrogen and carbon chemistry, either in the form of catalyst or co-catalyst. So I would like to dedicate this presentation to my sir, as I mentioned before, Professor Dr. Ashoka G. Samuelson, on his 65th birthday celebration. I won't call it as a superannuation because his knowledge always will be serving to many people, enlightening them in organometallic chemistry. So he is not going to superannuate from his teaching and his research work. So therefore, I'd like to call it as it's a 65th birthday celebration. When I joined in his group in 1997, Professor told me to make some compounds to study the copper-copper interaction. In this regard, I could make trinuclear copper complex, few of them complexes, stabilized by diphosphine. And in this case, we could encapsulate perchlorate anion. And this perchlorate anion binds with all these three copper center in unequal manner. And therefore, we call this complex as making and breaking clusters. Later on, Ritu Ahuja, she made one dinuclear complex in which two copper center contains two different coordination number. One is a 4-coordinated one and another copper is a 3-coordinated one. And here professor asked me to think about this, why one copper is taking 2 acetonitrile and another copper is taking only 1 acetonitrile, although she could make this complex in acetonitrile medium. So here he proposed that the 4-coordinate complex can act as a electron donor, and the 3-coordinate copper center can act as a electron acceptor center. And therefore, here one can see there will be a donation and acceptance character involved in this particular complex. With this, we started doing some DFT calculations. In fact, I could not succeed in this by getting a straight line. In fact, when I left Professor lab, he told me, Shivashankar, you should get a straight line, see it goes up again. Because after 2.4 Armstrong unit, it should get stabilized. So somehow I could not get as he expected. But one important thing here, what we noticed, at a 2.4 Armstrong unit, the copper, this particular dinuclear complex, gets stabilized. In fact, Ruthenium, when she got a crystal structure, she also found that the copper-copper distance is about a 2.4 Armstrong unit. So there was a very good correlation between experimental work and theoretical prediction. And these are the things that he likes. That's the reason why I put it these things. Then later on, Christina, she prepared the hexanuclear complex. In fact, Dr. Yanan Murthy also mentioned about that, that hexanuclear complex. When she tried to add a zinc chloride to that, she could get a carbene. I think by the time she left. So professor told me to check, whether it is a carbene or not. Then we studied that reaction, and later on we simplified the scheme just by taking copper acetonitrile and phenyl isothiocyanate and our required R group we introduced, R minus in fact it is. Zinc chloride also, that's a metal center that we used. Then we could get the carbene, which is spectroscopically has a similar feature with what Dr. Christina reported in her thesis. And later on, we simplified this process and we could make so many of copper-1 carbene complex. In fact, I had almost 13C carbon NMR in my hand. That time we noticed Professor Roald Hoffman from Hoffman from Heidelberg University. He published on Angewandte chemie paper mentioning that the first 13 carbon C NMR of isolated copper-1 carbene complex. Somehow we could not get a good credit as we expected for this particular work, but I think Professor also he likes this particular work very much. Then later on, we had one question like the copper is hard or soft. In fact, Dr. Bera, in his thesis also he could do some work related to this. Then I was asked to check with some more examples to find out whether copper-1 is really soft or hard. Because we always think and also we see in the textbooks that the copper-1 is more of soft in nature. But in this case, we can see as we increase soft coordination, that means by adding sulfur coordinating ligand then H2O, the net stabilization energy which goes down. That means the copper-1 prefers soft ligands. And also, there should be at least 1 or 2 hard ligands around that. That means the copper-1 is not fully soft. Again, it doesn't take 4 of them or water to be around. And thus, it also tells that it's not a hard metal center. And thus, we can say probably it's a borderline case. So these are the things that that I could do in Professor AJ's lab when I was a student. So now I am getting to my the Pondicherry University, my independent work, what I did here. Before getting to that, I would like to thank SARB, CSR, and UGC for funding, and also Pondicherry University for the facilities, and all my students. So this is the overview of our laboratory work, and we always use experimental work and computational work to design new chemical systems, and those chemical systems we want to explore into at least 3 different areas. One is developing new organic molecules or methodologies, and another one is bioorganometallic chemistry. Here, carbon monoxide releasing molecules and anti-cancer and antidiabetic type of work that we are interested in. And the third thing is the industrially important work, something like conversion of dinitrogen to ammonia. In fact, in this area, we progressed a lot, and we have our own methodology how one can bring nitrogen and dihydrogen together in order to make ammonia at room temperature. In fact, theoretically, we succeeded. Experimentally also, we isolated one of the intermediates, and that work was published in the European Journal of Inorganic Chemistry on the cover page article. And of course, conversion of hydrocarbons into alcohols and fixation of carbon dioxide into acids. So these are our some of the interested work in our laboratory here. In fact, wherever I went, even to Germany or US Scripps, I was told to work with experiment as well as theory. That privilege I got. In fact, this training I got it from my professor AGS. In fact, he taught me theory and also experiment. At one point of time, I did not want to do any theory because you know, those days the Gaussian, we have to write a Z matrix and all the things and the proper symmetry and all, we have to get it. I had a tough time in getting all those things, and Dr. Bera used to help me. The time what happened, I could not get a tetrahedron geometry for copper 1 complex, particularly when we put a 4 water molecule or 4 HDS ligand. So one day I was walking in the corridor and the professor came across and he asked me, Shoshankar, what about your calculation? I said, sir, it's running, but I'm not getting that thing. Therefore I stopped. No, don't stop. You continue like that. Then he encouraged me, in fact, in several ways. Not only chemistry, a lot of things he taught to me. Truly, I am grateful to him. Today, what I am, it's totally because of his wonderful guidance. And plan of my presentation, although I put many things here, because of lack of time, I can restrict to 20 minutes. So I try as much as possible to present this. The copper as a reducing agent in nitrogen chemistry, first I will discuss about that, and then a carbon insertion reaction catalyzed by a copper complex, and the copper catalyst the synthesis of sulfoxyl helides as a room temperature carbon source. Because diazo compounds, we all know that there's we have to store it in the refrigerator all the time. And most of the laboratory, whenever they need it, they have to make it and use it. Whatever left over, they have to store it. Diazo compounds are, most of them are not commercially available. But the sulfoxyl halide serves as a carbon precursor. We all know that Core reagent, so the CH2 halide of sulfoxyl halide. So here what we have done is sulfoxyl halide we made in so many grams and also so many examples as a room temperature carbon source. So we can make it and put it in the bottle, keep it in the shelf. We can still make it commercially available also in this aspect. Copper complex catalyst the reductive coupling of diazo compounds also I will be discussing, and finally conclusion. And this particular dinitrogen related work was carried out by Bhaskaran. So in fact, this was my first independent paper after I took up a position in Bharathidasan University in the beginning, from when I returned from Scripps, Professor Roy Perena's lab. So at the time, I just was given 2 computers, so we thought of to take up some computational work to start with. So Dr. Bhaskaran, that time he joined with me as soon as he completed his MSc from VIT University. And then he worked on this, and you can see here the title, the historically significant Ruthenium complex we mentioned, and we also mentioned that a new life for an old complex. And this paper was dedicated to Professor Ashoka G. Samuelson on the occasion of his 55th birthday. In fact, I wanted to dedicate this. Then after acceptance, the editor told me, no, you simply cannot just say dedication. You have to mention the reason. So I found the 55th birthday of him, and therefore I just put it to 55th birthday. That is the, otherwise this the 55th is this reason is for is this. Okay, we all know that Helen and the son of dinitrogen complex. This is the first complex reported in the literature where we can see dinitrogen is coordinated to the ruthenium. And this complex, when they reported the crystal structure and all other things, soon after that they also reported that the dinitrogen can be functionalized. Little later, Chart group from Great Britain, University of Sussex. So he carried out some control studies, and then they said that sodium borohydride that reduces the impurity, which is actually hydrazine. That is the source for the ammonia, not the dinitrogen. And therefore, the corrigendum, they mentioned that dinitrogen in this complex is really not reactive, and only the hydrogen impurity undergone a reduction in the presence of sodium borohydride and produced the ammonia. And this hydrogen they used to make a dinitrogen in situ. That means oxidation of hydrazine can produce a dinitrogen in situ, and that dinitrogen can form a complex with ruthenium. That is the story about that. Because I'm not going to put much of introduction here because of the lack of time. And then we asked the question why the dinitrogen in Hallan and Sennev complex is not reactive towards the protonation and the reduction. And then if at all if you want to make it, then how to make it? These are the questions that we raised. And then we, as I mentioned before, I was given a computer, so thought of make use of it and started doing some calculation. We all know that the Chart type of complex, that is 6th group metal ion stabilized by diphosphines with 2R1 dinitrogen coordinated system. You can see here, I'm sorry about I could not put the chemical structure here. So here you can see the formula of that molybdenum dinitrogen diphosphine complex. And this complex is known to undergo protonation and reduction. At the same time, the short paper, the science, and also there was on Jack's paper. They mentioned that this triamidoamine complex of molybdenum dinitrogen can undergo reduction and protonation, followed by. In fact, it follows a Chart type of mechanism. We all know what is a Chart type of mechanism. And it can produce ammonia in a catalytic manner. In fact, a turnover number, if you see, just a maximum of 4 or 5. That's all it is. But the lesson is these two compounds can produce ammonia. So now what we thought, let us compare the electronic structure of the Hallan and Sennev complex with all these known compounds which can produce ammonia. And then we could see here that the contour plots, the first one, ruthenium Hallan complex. Here, the HOMO is located on the metal center. LUMO also is located on the metal center. Okay. Therefore, when you add a proton, it can bind only with the metal center. It cannot go to the dinitrogen. Whereas in these two compounds, you can see the HOMO is located on the metal center, as well as there is a contribution from dinitrogen. That is a beta nitrogen of dinitrogen. And here, the DPP complex of molybdenum also you can see. The HOMO again is located not only in the metal center, there are also contribution from beta nitrogen of coordinated dinitrogen. And thus, these two compounds can undergo protonation, whereas this particular Hallan and Sennev complex cannot undergo protonation. Now, if we go for a reduction, again, the LUMO here, you can see there is a contribution from dinitrogen, and Chart complex also there is a contribution from dinitrogen, and thus when you add an electron, it can be distributed to the metal center and also to the ligand center orbitals. Whereas in this complex, it can go to the metal center. So either a reduction or protonation, it won't affect the coordinated dinitrogen of ruthenium complex. Whereas in these two complexes, very well they can undergo any type of reduction or protonation reactions. And thus, these two compounds can undergo protonation, and this one cannot undergo protonation. If you look at the charge distribution over dinitrogen and also the metal center, you can see here the metal center here carries a 0.566. This is a natural population analysis only, and alpha nitrogen carries a minus 0.7, and beta nitrogen again 0.16. Whereas if you see other rest of the two compounds. Here, I want to get your attention on the N beta. Now you can see here, there is a negative charge. Here also there is a negative charge. Whereas here, there is a positive charge. I think this is probably the experimentalist may say there is no difference at all, because this is a 0.16, 0.02, 0.01. These are all nothing. But these are that's all the difference that we could see. And we are not telling this would be totally responsible for why the dinitrogen in this complex is not reacting. But this could be one of the reasons. Okay, this could be one of the reasons. So here these two systems can undergo protonation, whereas since here you see more of a positive charge, it may not undergo a protonation. And therefore, now you can see these are the commonly used protonating agent in this nitrogen chemistry. Now lutetium, if you use, you have to pay 69.12 kilocalories per mole, delta G it is. And if you take any acid in water, then 35 kilocalories per mole you have to pay, and just for water 130 kilocalories per mole you have to pay. And therefore, none of these reagents should be suitable to protonate the halon and sennit complex. But these are all works for these two compounds. That is a short complex as well as a short complex. Then what we thought, let us reduce it and see. So we have taken this complex and added electron to this system and make it to +1. That means a ruthenium +1 complex. And now you can see the electronic structure here. Now the dinitrogen almost is getting a hydrogenic character. And noticeably you can see here there is a contribution from a dinitrogen to this HOMO. And thus when you add a proton to this, probably it can react with the dinitrogen. Although you see the contribution from metal center, but when the proton reacts with the metal center, it should oxidize the metal center. And also here you can see the coordination number of ruthenium in this case is saturated. That means maximum 6 is reached. So addition of proton on the metal center is less viable. We checked all the things by computational methods, but the most viable pathway is adding a proton to the dinitrogen. And thus, so what we are saying is here, after reducing this, now it is very obvious that you can see the improvement of negative charge on the N beta. In the previous case, you can see 0.16. Now it has improved to minus 0.14. And thus, probably now the N beta, that means beta nitrogen, can uptake a proton by using any of this listed out protonating agents. And now the reducing agents, if you see, these are the things that we used. Copper, that's the reason I just mentioned in my title also. Copper can be used in nitrogen chemistry. So you can use a copper powder as a reductant here, and the cost is almost 1.43 kilocalories per mole. Still, it can take place very well, but magnesium is superior than any of these reagents. So magnesium, in the case of magnesium, you can see just minus 12.97 kilocalories per mole. And other decamethyl chromazine and all, it's improved to 20 kilocalories per mole positive. So it may not take place. Again, probably one can ask this question. In the synthesis of ruthenium complex, they used sodium borohydride. But at the time it did not undergo reduction, because now you can see in the case of sodium borohydride, the reduction will cost plus 62.21 kilocalories per mole, and thus the reduction is not possible by sodium borohydride. So either you have to use copper or magnesium. Magnesium is the suitable one, but still one can use a copper. So using these two reducing agents and any one of these protonating agents, now you can see the protonating agents. In the case of lutetium, you have 5.35 kilocalories per mole, acid in water minus 28, and just water minus 27. So you can use any of this, one of these reagents as a protonating agent, copper or magnesium as a reducing agent, and add to the halon and sennit complex and see whether you can functionalize the dinitrogen or not. Now this is the simulated mechanism for this halon and sennit complex. Here I am not going to discuss the whole thing here. I just want to mention here one thing. The ruthenium 2+ complex here, first we have to reduce it to make it to +1 complex, and then you add a proton to functionalize it. Whereas if you see short mechanism, always he proposed the protonation is the first step, not the reduction. So what we say for halon and sennit complex, instead of taking a protonation as the first step, you reduce it and then try to functionalize it. It should work as per this calculation. So these are the energy barriers. Now you can see, I'm not going to detail of this also. You can see here 13 prime. You can reach this place very well without having much difficulties. There are so many ups and downs here. But whatever be the case, at the maximum of almost 20 kilocalories per mole only the barrier you can come across. and therefore 13 prime is very much feasible to access it. But recycling it is difficult because when you go from 13 prime to 1 prime, you have to pay a lot of energy, and therefore recycling this is not possible, but at least you can functionalize it. In fact, one of the short review articles also, you mentioned that Hallan and Sanif complex is historically important complex, but chemistry-wise is not useful. But in this case, we just thought of to say historically important complex, to some extent it can be useful, not fully not useful. That's what in the conclusion also I mentioned. The second part of my work is a carbon-related work. Here, I just want to talk about only one thing because there is no time. The copper-1 complex, what you see here, that's what we synthesized, and the Ramakrishnan and Febin, they involved in this particular work. In fact, this paper we dedicated to Professor A.G. Samuelson on his 65th birthday. And here, when you put amine, alcohol, and acid olefin to one particular substrate, can you get a chemoselective reaction? That is the question we asked. With this objective, we have taken a diazo compound and used copper acetonitrile. In fact, it's a very famous in Professor's lab. So we use that same copper-1 salt, and now you can see the carbon is undergone insertion into NH bond as well as OH bond. And if you use amino acid, again you can see there is insertion into NH bond and also OH bond. Okay, so copper-1 acetonitrile can do this much mixture of product always. And then this is the objective of our study. That's what I mentioned before also. If you can make any catalyst and do this chemistry in a chemoselective manner, we can get more benefit. So with that, we made this complex, and you can see the crystal structure of also of this copper-1 complex. And then we conducted the reactions, and we got 69% of NH inserted product. There is no OH inserted product in this case, but we could isolate some of the carbene dimers. And then we optimized the reaction condition, particularly the solvent here. Anyway, the methanol is a suitable solvent. We use a methanol, but the carbene did not undergo insertion into OH bond of methanol in the presence of our catalyst. That is a very important observation in this reaction. And here also we screened a lot of copper-1 salts, but our complex is superior than any other copper-1 salts what we used here. And a lot of examples we could make. Anyway, we don't have time, so I'm just showing you the overview of our results. And the amino acid also we used. Here also we could get only an insertion into NH bond, not to the acid OH bond. So we could make so many examples, and we could get. In fact, here phosphonates also we could introduce, and these are all somewhat biologically important systems. That's what. That's why we could publish in JOC at the time.
Can you just, Shivashankar, can you just?
Yes, sir.
Can you just hurry up?
Okay, sir. I will complete.
Sorry to interrupt.
Okay, sir. No problem, sir. Thank you, sir. Thank you so much. And this is the proposed catalytic cycle. I will stop with this catalytic cycle. Actually, why we could get a chemoselective reaction, particularly the carbene undergoes NH insertion, not COH or OH insertion. To understand that, we simulated this catalytic cycle also. We have taken the copper-1 complex of ours, and we made a complex with this particular organic substrate. And this one is NH coordinated, and 1A prime is OH coordinated, and 1A double prime is acid coordinated one. Here, the 1A prime and 1A double prime is energetically very high when compared to 1A, and therefore 1A is more stabilized one. That means amine coordination is a preferred over OH as well as COH coordination. After getting this 1A, when you add a diazo compound, it can form a carbene and then further they can undergo nucleophilic addition and get this type of system, and finally, you get your expected product here and regenerate the catalyst. So here I use the concept of what we developed in process lab. There, copper one is not soft; it's a borderline case. So that element of information we put it here. This copper one here is stabilized by 2 phosphines, and also it prefers 1 nitrogen. Nitrogen is somewhat less order than OH, and therefore it doesn't mean the copper one doesn't seek for a more hard coordinating site, but it could be a moderate one, and therefore it prefers amine over OH and COH, and thus this 1A is more stabilized than anything else. Therefore, we could get chemoselective organization. These are the things, and anyway, all these things we don't have time, and this recently, this particular, this alone I will show. In fact, I discussed this particular catalytic cycle also with my sir last time when I visited him, and this is under revision in applied organometallic chemistry, and this also we dedicated to him for his 65th birthday. And anyway, so these are the things, and copper we used for reducing as a reducing agent, nitrogen chemistry, copper in chemoselective reactions, copper in making phosphonates, and the copper is in synthesis of sulfoxyl halides, and copper also we used in reductive coupling of diester compounds. So these 3 things we could not discuss because of lack of time, but I hope I convinced you, as I promised in the first slide, that I could show you some of the copper mediated reactions. So I thank you again, and especially I take this time to thank you, sir, for helping me in many ways, and you are the one who taught me the organometallic chemistry in depth. I just want to mention only one small thing, just 2-3 minutes. Sorry, a few seconds. Actually, sir, never he gets angry when we go with some wrong results. Actually, whenever we, as a student, we always overinterpret certain data. So whenever I did so, sir used to tell me, Sivashankar, this is not wrong. Like, he never told like that. He told me always, Sivashankar, go and get a proton number, go and get the carbon number for this, go and do this particular experiment and see. So after I go back and do all those things, and I myself used to realize, I was wrong. So sir never told me you are wrong, directly like that. He always made me to realize it, that I was wrong in doing certain things, and so many ways sir helped me, and truly I would say what I am today here is all because of my sir. Thank you, sir, and thank you all for listening to me patiently. Thank you.
Oh, thank you very much, Professor Sivashankar. The talk is now open for discussion. 1 or 2 short questions, because are there any questions?
Durga Prasad has a question.
Durga Prasad, please.
Professor Sivashankar, a very nice presentation. So you talk about this highly chemoselective of NH insertion to the copper carbines. Yeah. So my question is, you often use this donor acceptor carbines. Do you observe the same kind of reactivity when you have an acceptor diazo compounds or a single acceptor diazo compounds?
We used a single acceptor diazo compound because aryl group you can treat it as a somewhat less electron accepting group, and therefore probably I think we used only acceptor to some extent.
Okay, so how the chemoselectivity changes with that? Do you observe the good chemoselectivity, or you see some kind of OH insertion?
Actually, we used the indium catalyst in some other occasion. There we could get OH insertion then over NH insertion. But in copper case, particularly with this substrate, we could get only NH insertion.
There is a lot of debate on the whether the copper 1 or copper 2 is involving in this kind of reaction. So what do you, can you comment on it?
Actually, we have not done any in situ spectroscopic studies to completely comment on this, but still, we believe it's a copper 1 is responsible for this. Because if it is a copper 2, probably we could see some OH inhibition also, because copper 2 prefers OH binding rather than NH binding.
Okay, thank you.
Yes, thank you.
Okay, we will leave it at that, and further discussion can be had later. Now I leave the floor to Professor Sebastian to take over the chairmanship and conduct the remaining part of this session. Thank you.
Thank you, Professor Krishnamurthy. Let me start by thanking the organizers for giving me this opportunity to chair this particular, to co-chair this session, which is in honor of Professor Samuelson, who has been a very dear colleague for a rather long time. The first speaker actually is Professor Akshay Kumar, who is from IIT Guwahati, a former collaborator of Ashok, and he is going to talk to us on synthesis of specialty chemicals via catalytic transformations by pincer metal complexes. Professor Akshay Kumar, are you ready? Please start.
Yes, sir. Can you hear me, sir?
Yes, I can.
Yeah. Good morning, all. It's a very special occasion, and my heartfelt is the full slide mode visible now?
Yes, it is.
Okay, fine. Thank you, sir. So I have been very fortunate whenever I see sir's photo, and like when I see IPC, when I see IIC, lot of memories churn, and then it makes me overwhelmed with feelings. And what I can do, like I'm short of words. I can just tell thank you, sir. Thank you, sir, for providing me such a very invaluable platform for my research career, for enhancing my knowledge on organometallic chemistry, for giving me the basic training, and not only that, for giving me a set of seniors, colleagues who have handheld me all through my academic career so far, starting from Rajshekhar, Ritu, and Rakesh, especially to mention Rakesh, like I have learned a lot of techniques from him, and I had good rapport with Sangeeta Das, and good interaction with Durai Pandi, Raja Mitra, Arun Kumar, Tenraj, and so on. And not to forget, the best thing was all students of AGS sir have sort of helped me in one way or the other. Not to forget the help at various stages I got from Professor Jitendra Bera sir, Nasim Murthy sir, and at IIT Guwahati, the continuous interaction I have with JBB sir, they have sort of helped me move forward in my research work. So these are some of the photos that I derived joy from. It was a rare occasion when I was very fortunate to have Sir and even Alan, both of whom are thorough gentlemen, and getting training from them is a blessing indeed. And I also thank all the faculty members of IPC department, my teachers, Professor Jagirdar sir, SS Krishnamurthy sir, Ramkrishnan sir, P.K. Das sir, Mukesh sir, Tilagas sir, and I think I have to move on with my talk now. But then I just thought that I would share that I had a special moment with the SSK sir at IIT Madras when he handed over the MTIC Best Poster Award. These are all special moments, and they sort of continuously motivate us to do chemistry. And this is a preamble of what I do and what I plan to do. We are looking at the transformation of waste to anything useful, and that I do with the help of organometallic catalysts basically pincer catalyst in generation of specialty chemicals, fuels, hydrogen, and a small portion of my work is also dedicated on the generation of fluorinated molecules for optoelectronic devices, sensors, and even optically transparent materials for space applications. Way long before I joined Alan's lab, the first concept of pincer was sown in my mind when AGS sir gave a literature search talk on pincer complexes in our lab in 2005. That was in the conference room, IPC conference room, top floor, probably where some of the faculty are all sitting now. And there I learned that it is in contrast to bidentate complexes and multidentate complexes, pincers have a very unique feature in which you can attain optimal control on reactivity and on stability, and the control is multifold because you could either control the electronic factors, you could control the chirality, you could control the trans-influence effect, and the ancillary ligands would determine whether you could generate catalytically active species or not. And then the polar groups, they are very good candidates, not only for controlling the catalysis remotely, but also to anchor them on heterogeneous support, so that we could have catalysts now which have selectivity signature of molecular catalyst, and in addition they are blessed with the recyclability that is signature of heterogeneous catalyst. So in last time I gave the talk in IPC, I told that I had synthesized a class of pincer metal complexes, NNN based. I told that time that contrary to the Goldman's case, wherein it was a PCP with a strong trans-influencing ipsocarbon and a pi-accepting phosphine, the nitrogen here is weakly pi-accepting, and also it is weakly sigma donating compared to the amine. And then in contrast to Milston's system, these are weakly pi accepting as well as weakly sigma donating. And in contrast to Goldman's system, this nitrogen has a lower trans effect. And then this is a typical 18 electron species. Ruthenium is having 8 electrons and 6 are coming from nitrogen, and then 2 each from the chlorides and 2 from phosphine, making them easy precursors for 18 electron species. That time I had also explained how we had screened the atom transfer radical additions. I'm not going to explain in detail, but then this was one of the first reports where we got about 5,900 turnovers in the atom transfer radical addition of styrene to carbon tetrachloride to styrene. This we had even extrapolated to the N-alkylation reaction, wherein starting from the alcohol, we had generated the base, requisite base in situ, and then used it for N-alkylation, which again I had discussed in my talk in IPC the last time. Today, what I'm going to focus is on one reaction, and that reaction is the transformation of waste glycerol to lactic acid. But then in the meantime, we thought that in addition to having PPH3, which PPH3 is sort of toxic, and then though it is very easy to follow catalysis with the phosphorus NMR, but then it is the thrust is now to move away from phosphine based ligands to phosphine free ligands. Therefore, what we did was we took these ligands and treated with paracymene in the presence of carbon monoxide to generate this complex. This complex itself is very satisfying, because then this is a very good example of a typical organometallic complex. Again, this is 18 electron species, wherein we have varied the R groups to have a control on the electronics, and then the control on the sterics we have we have synthesized more open bisimidazole-based pincer complexes. Now, I would go straight away to the work that we now have access to a large library of pincer ruthenium complexes, and that complexes, we have used it for the transformation of glycerol to lactic acid. Now, why glycerol? Glycerol is highly abundant, not only because it is obtained from certain fermentation processes, but also because with the moving away from fossil-based fuels, the focus is now on biodiesel, and biodiesel glycerol is an inevitable byproduct. And globally, the overall glycerol production is about 2 metric tons in 2004, and this is greater than 5 million tons in 2019. And this glycerol actually is a good source of lactic acid. And the lactic acid market is good, but in India, still we don't have a good market of lactic acid. But globally, particularly in US, the lactic acid market is very good, and the market rate is also unprecedented. And the byproduct of this reaction is hydrogen, and we need not tell anything about the clean burning hydrogen. There are a lot of heterogeneous catalysts to do this, but then the problem is the selectivity. They give lower yield of lactic acid, and getting a stereoselective fashion, lactic acid in a stereoselective fashion is a challenge. Homogeneous, there are a handful of homogeneous catalysts, but then the mechanism is not properly understood. But then there are very good catalysts the first report came from Crabtree and then by Beller, and these are all very good catalytic systems. But as I said, they are either based on precious metals or based on phosphine ligands. So considering these variety of applications of lactic acid, we wanted to see if our phosphine-free catalyst can accomplish this transformation. We have done a lot of reactions with the phosphine as well as phosphine-based phosphine-free catalyst based on carbon monoxide, but then for the sake of this talk, I have picked 4 catalysts so that I can explain this reaction both from synthetic as well as mechanistic point of view. So here is the reaction. We have glycerol. We treat it with base, 0.5 equivalents in the presence of ruthenium catalyst, in the presence of ethanol, under in an open vessel with the argon flow. And what we see is that we get lactic acid and a small fraction of formic acid, ethylene glycol, and hydrogen. Now, as we look at various catalysts, you see that with T-butyl-based bis-aminopyridine catalyst, you get fairly decent yields of lactic acid and a very small fraction of ethylene glycol. The selectivity is very good. Whereas with the phenyl catalyst, the yield is less. On the other hand, with the sterically more opened bisimidazole catalyst, either with the chloride anion or a PF6 anions, the yields are comparable but higher than tertiary butyl. So there are three things to note here. The first thing to note is these two catalysts are likely to generate the same active species, and the phenyl is the gives the very poor activity. So before I go into the mechanism, I wanted to say that we have optimized this. We have optimized this such that we get good yields of lactic acid, 90% lactic acid with 98% selectivity, and so 92% is the so for example, this ethylene glycol and formic acid, the percentages are 1%, and the conversion of glycerol is about 92%, and the selectivity towards lactic acid is 98%. So the selectivity is good. That means we are getting exclusively lactic acid. Not exclusively, it would be cheating to say that. But then at a very lower loading of catalyst and in the presence of water, we get about 44% with 96% selectivity. Turnovers are higher. Okay, now let us look into the mechanism. And we do computational studies, but then we get it validated. We used to get it validated by Dr. Hemant Kumar Srivastava, a dear friend of mine, I'm very sorry to say that he succumbed to COVID 2 months back. He was a very nice person, a thorough gentleman. So this, regardless of whether this is a bis-imino-based pyridine catalyst or a bis-benzimidazole-based pyridine catalyst, both of them would generate this dialkoxide species. In the case of bis-aminopyridine catalyst, it is loss of PPH3 and then a salt metathesis with potassium hydroxide and glycerol. In the case of this cationic complexes, it would be loss of PPH3 followed by a substitution and salt metathesis. Both of this would give you the dialkoxide species. For the sake of convenience, we have represented only this pincer ligating fraction. So what is the mechanism? The first step is the beta hydride elimination to give the glyceraldehyde and to generate the ruthenium hydride species. This ruthenium hydride species can now undergo a sigma bond metathesis with glycerol to give hydrogen, which is being detected by GC, and give the give back the catalytically active species. So if you look into this, the entire transformation of glycerol to lactic acid, only the formation of glyceraldehyde is catalyzed, whereas the transformation of glyceraldehyde to lactic acid is a organic uncatalyzed reaction. It involves dehydration to give this, which undergoes tautomerization to give the corresponding diketone, which undergoes an intramolecular Cannizzaro reaction to give lactic acid. Now, if this is so, then the difference between the catalyst is mainly because of the difference in this dehydrogenation cycle. So to know that for the dehydrogenation cycle to be activated, we should generate the active species by dissociation of PPH3. What we found was with the T-butyl catalyst, the ruthenium phosphorus interaction energy was about 39.82 kilocalories, whereas in the case of bis-imidazole, it was 39 kilocalories. In the case of phenyl, it was 44 kilocalories. So the catalyst in which the PPH3 is the most difficult to be dissociated of to generate the 16 electron active ruthenium species is giving the less yield. So that makes sense. But then catalyst T-butyl 2-NNN ruthenium and bis-imidazole ruthenium are having similar bond energies. Then why is this difference? This difference is more pronounced because at lower loadings we get 90% lactic acid yield. So comparable bond energy, why is this difference? So when we looked into the mechanism, we saw that in the first step, the beta hydride elimination is highly kinetically unfavored for the T-butyl catalyst. Not only that, if you look at the formation of glyceraldehyde, it is so this is within the catalytic cycle. I'm saying this is more downhill. So this is 16 kilocalories, 31 kilocalories, and if you look in the subsequent step, which involves the sigma bond metathesis, you see that in this case, this is about 29 kilocalories here uphill. The kinetic barrier is about 29 kilocalories, whereas in this case, this is about 38 kilocalories. So, this is about 22 kilocalories. This is about 22 kilocalories. So, for the T-butyl PCP, T-butyl NNN ruthenium catalyst, the rate determining step is the beta hydride elimination, whereas for the bis-imidazole catalyst, the rate determining step is the sigma bond metathesis, or the hydrogenolysis step is the rate determining step. Now, if you compare this, even the rate determining step is much lower than the rate determining step of T-butyl catalyst. And the other thing is, if you look into this, the formation of glyceraldehyde is more exothermic. So it sort of drains out the glyceraldehyde, siphons out the glyceraldehyde in this step. And then, since this is an open vessel, the loss of hydrogen is more favorable here. Though this is a relatively uphill process, since it is an open vessel reaction, hydrogen is easily lost and it gives the product. So both the rate determining step as well as the beta hydride elimination, it is more favorable for the bis-imidazole based catalyst. And with bis-imidazole, you would expect that the electron density on ruthenium is less, which actually sorts of favors the beta hydride elimination. But then sterically, if you look at it, the reaction is more favorable. In the transition state, the t-butyl catalyst is much more crowded in compared to the bis-benzimidazole catalyst. So all this factor work in favor of the bis-benzimidazole catalyst giving good yields of lactic acid. So what we have done is, on a lab scale, we have taken 10 grams of glycerol and we have got about 8 grams of lactic acid. We are in a process of sort of doing it on a bench scale now, on a batch reactor. But then this is the current status. And on the other front, what we have also done is, I will not explain in detail, these catalysts also find very good application in upgrading ethanol to butanol. So tetraethyl lead was a classical additive to fuels, but now people are more focused on ethanol. But then the energy density of ethanol is very less. On the other hand, butanol can be a alternate oxygenate, and this can serve as a fuel additive. And what we see is we get about 98% selectivity to N-butanol in this transformation of ethanol to butanol. How is it done? Ethanol is dehydrogenated by this pincer catalyst to acetaldehyde, and acetaldehyde undergoes a aldol condensation to give the corresponding alpha-beta unsaturated aldehyde, which is then hydrogenated back by this catalyst to give butanol. At longer times, we get higher alcohols, but then the selectivity is hardly compromised. At higher times, after 6 hours, we could still have 80% selectivity to N-butanol. So we have actually investigated this reaction even from mechanistic point of view. The reaction is first order in catalyst concentration. A sort of first order with respect to ethanol. We are still not happy with this kinetics. We are re-investigating this. And so these catalysts find good application in biofuel production also. So the term specialty chemicals in the title is a ongoing process. We are looking at transforming this butanol, which is a useful C4 fragment, to specialty chemicals. So there are other reactions that we have looked at. We have looked at pincer nickel catalyzed N-alkylation reaction, and we have also looked at very recently a computational work on pincer nickel catalyzed cyanomethylation reaction, wherein we see that weaker trans influence of central nitrogen atom is detrimental for the overall energetics of cyanomethylation, whereas having strong sigma trans influencing ipsocarbon is pivotal, and weaker sigma donating imine nitrogens are also pivotal, whereas strong sigma donor amines, they give less yield. Now we have recently reviewed this pincer nickel complexes also, and then since we had a lot of large library of pincer nickel complexes, we have studied this for cytotoxic activity, but we find that they are active against both normal as well as cancer cell. But the interesting thing is this works by disrupting the membrane. A lot more systematic study needs to be done, but then my primary focus would be on catalysis. The other 10% focus is on generating pi conjugated molecules for optoelectronic application. We have reviewed this recently, and then we are in the process of generating the corresponding fluorinated molecules for space applications. So as I said, I'm fortunate, blessed to have got the training from ATS sir and continue that training in Alan's lab. I have no words. I will ever remain grateful, and I would always have a sense of gratitude, and I would try to deliver my best. And I would acknowledge the funding agencies, BRNS, Reliance, And then very recently we have this project on ISRO for space applications, and more importantly starts from ISC Bangalore for funding my research. Their work I have not discussed here, and I thank all my collaborators, and I thank IPC department, I thank Arunan sir, I thank Tilagar sir for providing me opportunity to present my work here. Thank you very much.
Thank you, Akshay Kumar. Thank you, Akshay Kumar. We have time for 2 or 3 questions.
Can I ask one question?
Yes, sir.
Yeah, please go ahead.
Yeah, this is here.
Bera. Yeah.
Akshay, if you look at what is happening at the metal, basically you need the 2 ortho center, okay, and everything is happening there. So in this context, I was wondering why pincer platform is absolutely essential. Like I can provide the same 290-degree ortho site by using any other ligand. Okay, okay. The first, I'll tell you the question and then you can respond. That's a general question. The second question is that when you are doing this dehydrogenation, most of our I see 4-member transition state. Okay. Can you look for something better? Because my understanding that whenever you do this involving 4-member transition state, and then when you have solvent, probably you can go ahead and include some solvent, few molecules to at least in DFT to get a better transition state. My final question is that when you are showing this biomass and then you are taking alcohol or biomass derived product and then you are making them to some useful product, is there any effort to start the reaction with the biomass? Because you see the issue I have with there is a background picture of biomass, but you are taking a pure molecule. Okay, I would rather take, for example, we did some time back. If you take the cigarette, buy cigarette, use the filter part. That is actually a very soft wood. So you can like to do that. Take real biomass, treat it, make it softer. Is there any effort towards that? I know my questions are long, but we can discuss. If the time is not there, we can discuss offline also.
Yes, sir. So actually, with the pincers, we are actually aiming at certain highly endothermic reactions. But then, in addition, those will take some time. In the sense, they are ongoing, and I have not discussed that. But then, just to sort of survive, those synthesized complexes are also used for alcohol dehydrogenation. But then we see that in the traditional bidentate complexes, we have tested a few of them. They lose out of gas after some time. The pincers, the complexes that have synthesized, they are highly stable, and they are stable up to 300 degrees.
Okay. We'll discuss separately. Like my question is that I can provide a tetrahydrotate ligand, let's say tripodal ligand, okay, or this pyrazole borate type of.
Okay, sir. So this part,
you are going to offer some kind of orbital, the frontier orbitals are different in this system so that it is most active. That line I was like, see if it is there. I do not know.
So the pincer part I will discuss with you later. And in coming to this ethanol and glycerol, we have used industrial grade glycerol and we have used bioethanol to validate our result. But then using directly biomass, I think there would be secondary product because biomass would have cellulose, and so we have a plan to sort of directly dump the catalyst into biomass and see what are the range of products that we get. That would definitely be there, but then that would be from an application point of view. But then when we are discussing mechanism, a systematic study is probably not possible by just dumping the thing. But then we are doing that. We are doing that just to look at if our catalyst can be used to transform this biomass to random products. But then getting reproducibility there, those are some of the challenges, not only to biomass, but also to plastic. But then as a traditional organometallic chemist, where I'm looking at proper mechanisms, I think this is somewhat what I like to do. But then those from an application point of view, we will do, but then getting a hold on the reproducibility will take some time.
Yeah,
actually, one more question.
We have from Balaji Jagardar. Was there a question from Balaji Jagardar?
Yes, Sebastian.
Yeah, please.
Yeah, actually, very nice work and nice presentation.
Thank you, sir.
Yeah, just a very short question because of lack of time. You invoked lability of the phosphine.
Yes, sir.
Right, in one of your pieces of. Do you see free phosphine?
In the glycerol studies, we see free phosphine, and when we do this HRMS analysis, we do not see any phosphine adduct. This is in very contrast to our previous studies, wherein the phosphine adduct was
I think
Akshay,
I think I lost your audio for a few seconds. If you can repeat, if you don't mind. Yeah.
Okay. Yeah. So in the glycerol studies, we actually see the evolution of free phosphine, and also in the HRMS, we do not see the phosphine adduct. This is in contrast to our previous studies, wherein we found that the atom transfer radical addition, wherein we found that ruthenium Cl2 PPH3 itself was regenerated after the reaction, indicating that is the resting state. Release of phosphine is the resting state. But in the current case, what we find is we find free phosphine when we add glycerol, and also in the HRMS we do not find any phosphine adducts. We find various glycerol adducts, but not phosphine adducts.
Okay. Yeah. All right.
Okay.
Yeah,
I think that's all that we have time for. Let me thank Professor Akshay Kumar for a very interesting talk once again. We will pass on to the next speaker.
Thank you.
So the next speaker is Professor Gernot Franking from the Philips Universität Marburg, which is in Germany, and it is early morning there. Early morning meaning 8:40 in the morning.
I think it's recorded.
Oh, it is recorded. Okay,
Yes.
Fine. I didn't know that. So anyway, the Professor Franking is an expert theoretical chemist, and he works in the area of chemical bonding and valence, and he is going to talk to us on that particular topic, chemical bonding and valence. So we will now listen to Professor Franking. I don't know what about questions?
Yes, sir. I'll try to share this.
I don't know. So there are no questions. I mean, right?
Yeah. After the talk, there are no questions.
Okay.
You can send emails to.
Can you hear the talk?
No.
No, I think we may have to. We don't see it also. Maybe we should share the whole screen. Now we see only the list of files.
Okay.
Just share the screen.
Yeah, I'll do that.
With audio, I think we have. I think you may have to share the screen and audio. I think, yeah, we can see the presentation, but we don't hear the audio. We don't hear the audio of the buses. I don't know if there is a way to.
Oh, okay. Okay. Let me see.
Tested this.
Do you not hear the audio now?
No, we cannot.
We cannot hear the audio. It's
Okay.
we should test this. I think there is a way to share the.
Okay. Let me try once again.
In this we should have probably tested before because the audio
Okay.
doesn't come. So could we do something? Maybe we can upload the video file in the same website.
I don't have laptop. Once, yeah. One second. I have this speaker. Can you hear it now?
No,
can you hear it now?
No, we cannot. I cannot.
We don't see anything also now. If Leo has some comment.
Once.
Yeah, so while for sharing with the audio, when you're clicking on the share button, on the right of that share window, you will get an option of include computer sound. You have to enable that in order for the computer sound to be audible.
I shared the, yeah.
Yeah, just I don't have it. No. It's, I know there is an option.
Debashish, when you share it, there is a share video option also that you can check.
Share video option.
Yeah.
It's because it doesn't have any information.
One second, yeah. Mic in. Headset.
Include computer sound, it comes right there.
I can hear it, but
if you have a laptop, you can.
But it's a big. Okay, I'll share.
You have to turn off your audio, Devashish.
Yes. Okay. Can you hear? No. Okay, I'll share.
This is common.
Yeah. Oh, okay. I did not.
Include computer sound.
Speakers on.
Okay.
No, Devashish, you have to mute your microphone. Then only we'll be able to hear you. Hear the recorded lecture. Professor Samuelson, you are pinned.
It's a big file.
Because it's a it's done.
That. Yeah, I have given to Professor Mukesh and because I think he will share.
Let me see if I can. I think let's maybe I will The share button itself, are you able to turn the audio on the buses?
He is probably muted.
No, Devashish is in the committee room right now. I see him behind Ashok.
Can you hear now?
No, we hear Mukesh, you are speaking. I think you have to mute your microphone.
I'm trying to share because he sent that to me.
Okay.
If we keep the audio on, it might work, Mukesh. Just keep the audio in full. Keep it next to that. It might work.
Very weird.
Professor, no, I don't think that would work. What the website says is that you can join the meeting with audio off and share the video screen with the computer sound, like Shukriyo was saying a few minutes ago.
Include computer sound is there.
Yes, that has to be enabled.
Yeah, I have enabled it. Let me see.
Yes.
But you may have to switch off your switch off your audio, as in microphone and speakers both.
Thank you very much. It's a great pleasure.
Yeah, we do hear now.
Lecture
for Ashoka Samuelson on the occasion of this formal retirement.
Here now?
Because I'm sure
Now we hear, but the presentation is gone.
you belong to those people where I have both the highest esteem scientifically and personally, and the visits at Bangalore, and you have also been at Marburg.
So the presentation is okay. Now it's
been so rewarding for me, and
yeah, we see
I
both
hope
audio and video
in
in
person again in the near future.
That's
Since
fine.
I know that you have spent so much,
I can see it. It's coming in.
Now the remaining time on science.
Can you start it again?
Topic we can hopefully share in the future. I've done my best to concentrate on what we have been doing in the last 3 years, which is
From the
upgoing
beginning?
until now.
Okay. Yeah, now?
Yeah, it looks fine.
It looks fine. You can start.
So we have started recording. So
Yes.
Mr. Franklin, you can start.
Thank you very much, and it's a great pleasure and honor for me to give this lecture for Ashoka Samuelson on the occasion of this formal retirement, because I'm sure that he will never really retire. Ashoka, you belong to those people where I have both the highest esteem scientifically and personally, and the visits at Bangalore, and you have also been at Marburg, have been so rewarding for me, and I hope that we can see in person again in the near future. And since I know that you cherish science so much, I want to devote now the remaining time on science, the non-scientific topic we can hopefully share in the future. I've done my best to concentrate on what we have been doing in the last 2-3 years, which is upgoing until now, and this is the topic of my research. It's called chemical bonding and malacy. And I start by looking on the periodic system, and I recall that when we take the periodic system nowadays, we look on it and we divide it into the so-called S block, P block, that is the main group compounds, the D block and the F block. And I want to remind you that actually this distinction, which is usually associated with certain roles into the 2-8 electrons, or the 8-octal rule, or the 8 electron rule, actually was already suggested 100 years ago. And this was done in 1921 by Irving Langmuir, who wrote in Sciences the postulates the electrons in atoms tend to surround the nucleus in successive layers containing 2, 8, 8, 18, 18, and 32 electrons respectively. Irving Langmuir built up his rules on the famous 1916 paper by Gilbert Lewis, but he is actually the author of proposing these rules, which are still nowadays valid. And it's important to point out that this was done 100 years ago, 1921, 6 years before quantum theory was developed. So it's a rule which survived quantum theory. But it has, of course, been supported by quantum theory, but it should be done in modern ways. And how it should be done, I will tell you by jumping on it again. So here we have, still nowadays, we have the 8-electron rule, the octet rule for the main group elements, the 18-electron rule for the transition metals, and the 13-2-electron rules for the lanthanides and the actinides. And we now focus in my lecture on the 18-electron rules for transition metals, also because I know that, Ashoka, your work has been dealing mainly with transition metals. So when we go to the carbonyls, which can be considered as parent systems of the carbonyls, when we know now that it holds very, very nicely for the elements nickel, palladium, platinum, which have 10 electrons, and in order to get 18 electrons, they need 4 carbonyls in the tetracarbonyls. We know it. And iron, ruthenium, and osmium, who have 8 valence electrons, need additional 10. So they get it from the 5 COs. And chromium, molybdenum, and tungsten, they need 12 electrons on 6 COs. And indeed, the most stable one and the coordinatively saturated one are the hexacarbonyls. So there's the next one, which have 4 electrons, titanium, zirconium, and hafnium. They should be the heptacarbonyls. But surprisingly, until recently, very recently, they are not known, in spite of quantum chemical calculation in 2008 by Fritz Schäfer, saying no evidence for the heptacarbonyls for any of the 3 metals as has been suggested from these experiments, despite the fact that our theoretical studies predict them to be reasonable molecules for all 3 group 4 metals. Why? Well, here now our work starts. We go back into the periodic system, and for some time I'm working with a Chinese colleague at Fudan University. His name is Ming-Fei Zhu, and he is mainly working in low temperature matrix isolation and gas phase chemistry. And he had a very clever idea. He said titanium, zirconium, and hafnium most likely do not form those 7 COs because they are too closely packed. How can we get those molecules that they are not so closely packed that they are far away? Well, you could go to scandium, yttrium, and lanthan, which have 1 electron less, but giving them, making them the anions, then they would be isoelectronium. So scandium, yttrium, lanthan minus have 4 valence electrons at the neutrals, like the neutrals here, but because of these additional electrons, the radius should be much larger, they should be much wider. And then it might be possible to get those elusive heptacarbonyls. And he runs the experiments for this, and he got surprising results. Here are the experimental values, and indeed, he found the heptacarbonyls. He got measurement. But somewhat surprisingly, the strongest signals do not come from the hepta, from the octacarbonyls, with 8 COs. He also got them with Nona and Decker, but he could show that these COs, beyond CO, are just the second coordination sphere. They are not directly bonded to the metal. And the convincing experiment was done by running a competitive experiment of a mixed yttrium-mangan metal target, and only the yttrium carbonyl survived, which has 8, a little bit of 7, but with 8. So it is not 7, it is the octacarbonyl, which is apparently stable. Now, an additional information was the vibrational spectrum, because he has the experimental means of measuring the gas phase vibrational spectrum, and he got that. And he got only 1 signal for scandium, yttrium, and lanthan, which means that they have very high symmetry and they are strongly red-shifted. And that's what was the information which came to us. He said, Gernot, I do not understand it. Could you get to run the calculation? And we ran the calculation, we found indeed that the scandium, yttrium, lanthan anions, here are the bond distances, they form singlet structures, stable singlet structures with cubic symmetry, point group OH, and the associated triplets are significantly higher in energy. So what he observed was most likely the singlet, which are stable for loss of 1 CO, giving the 18 electron heptacarbonyls. That was surprising. The question was, do the 18 electron, does it not hold for this system? We also calculated the vibrational spectra. There was perfect agreement with the experimental calculation for all these metals. No doubt about it. We published it. He got really the octacarbonyls. Why? Well, here is the MO correlation diagram. Here are the metal anions with the 4 electrons in the d orbitals. Here are the 8 COs, the sigma and the pi, and the split in a cubic field into these orbitals. The S go to the A1 gerade. The D, as known, split into the E gerade and the T2 gerade. The T1 ungerade go to this. But there is in a cubic field one molecular orbital having A2 ungerade orbital, which take 2 electrons, but they do not have a valence electron which has A2 ungerade symmetry. With other words, we do have 18 electrons filling the valence shell of the metal. The 2 additional ones are ligand-only orbitals, and you have to look into the symmetry of the orbitals, which was not possible by Irving Langmuir in 1921, because there was no symmetry known and no quantum chemistry known. That means the 18 electron rule is valid if you look on the symmetry of the orbital. And that becomes visible if you plot the orbitals. And here they are. The HOMO is the E gerade orbital, and you see the D orbitals. Below is the A2 ungerade, and there is 0 coefficient at the metal, is only a ligand orbital. Below is the triplet degenerate T2 gerade, again of the d orbitals. Then you have the triplet degenerate T1 ungerade, and you can see very tiny coefficients at the p. So the p orbitals apparently play a little role. And below it, as the S and the S, you can hardly see it. If you look on the coefficients and the MO matrix, you see very, very small numbers. So it means that the interaction takes place mainly via the d orbitals, and the S and the p are less important. And this can be tested by quantum chemical calculations. I do not go into details of this. We use a method called EDA-NOCV, where we calculate the interaction between the bare metals, the anions in this case, and the CO. We get the total interaction energy, which breaks down into the main components as a powder repulsion, a very small amount of dispersion interaction, and then comes electrostatic, coulombic attraction, and orbital interaction. And if we take those two as a main component, giving 100%, you can see 1/3 comes from electrostatic interaction, 2/3 from the orbital interaction. But what is nice about the EDA-NOCV is that you can break down the total orbital interaction into the pairwise terms, and then you get the pairwise interaction. And you can see that the major component comes from the E gerade and the T2 gerade. The E gerade is the donation of the d orbitals, the back donation in the p, which provide between 80 and 85%. The donation into the vacant d is significantly smaller, but still important. Makes sense that it's less important because the metal is an anion. It's a weak acceptor. Then we have a small contribution from the polarization of the CO, and you can see that the donation into the S and to the P, this is really negligible. The bonding comes mainly from the d orbital, and this can be visualized, which is very nice. And I show it to you now. The color codes do not show orbitals; they show the area of donation where you have the direction of the charge flow from the red to the blue, and you can see from the red to the blue, from the metal to the ligand, or the opposite, from the ligand to the metal. Here you can see it's just a polarization from the oxygen to the carbon and so on. I show it to you only for completion. That means that we actually do not have heptacarbonyls, we should look for octacarbonyls. So Ming-Fei said, well, if this is true, then for titanium, zirconium, and hafnium, it should also be octa, not heptacarbonyls. Could you have a look how it looks for this? Yes, I said we can. And indeed, the octacarbonyls also for titanium, zirconium, and hafnium are minimum on the potential energy surface, have singlet structures, are cubic OH symmetry. The heptacarbonyls and the hexacarbonyls, you can see it here, are also minima. These are the bond dissociation energy of one CO of the hepta and the hexa. And for the octa, I show it in a moment, because we see that thermodynamically only zirconium and hafnium should be observable, but not titanium. Although it is a minimum, it has a small barrier and then going to the heptacarbonyl. So we told him, if you run the experiment in the gas phase, you might find signal for zirconium and for hafnium, but not for titanium. And this is exactly what he found. Here's the infrared spectrum in the gas phase, and for zirconium he found for the neutral and for the cation. For hafnium, he found it for the neutral and for the cation he studied. But in case of titanium, he found only for the heptacarbon, not for the octacarbon. So we also compared the experimental and the calculated structures. That was published last year ago. Very good agreement. Most important, not for the absolute values, but for the frequency shift of the isomers and also of the COs with regard to them. That's a very good agreement. You can check all the data there. And if you make a bonding analysis, we find again the main bonding comes from the d orbitals, but now the donation into the d's has a little bit higher contribution, because now we have neutral atoms, not anions, and the neutrals one are slightly better acceptors. So the missing link of the group 4 metals have been found. For hafnium and zirconium, the octacarbonyls. For titanium is the hepta, only for steric reason. This was the important information which we got here. Now I skip some papers, some work, which got us to dare something, to do what should be impossible. And impossible means that the D block elements go until here. They stop here. Here are the S block elements. They cannot do D block chemistry. That is a rule. Is the rule valid? No, not for calcium. Ming-Fei, after some calculation and some work, which I skip here, ran the calculation, and he found, very surprisingly, even calcium can get an octacarbonyl. And here are the calculation for calcium, for strontium, and for barium. They also have cubic symmetry, OH point group. But now they have triplet electronic ground states. They are quite stable with regard to loss of one CO. And why do they have triplet ground states? Well, it's very simple. Because if you now look on the orbital correlation and diagrams, the metals have only 2 electrons. And in the cubic field, the HOMO is a degenerate A grade orbital, and due to Hund's rule, of course, you have one orbital electron occupied each orbital. They have a triplet. Apart from this, the correlation diagram is exactly the same. Now we have 18 electrons. 2 electrons occupy the A2 ungrade. So we have actually 16 electrons which occupy the metal orbitals, but 2 of them only singly occupy. This was really a sensation. We run the EDA NOCV calculation. We got again calcium, strontium, and barium bind via d orbitals. This was unheard of. But for both the neutrals and for the cations, we did experiments and the calculation for the anions. In both cases, we found that they are stable species. The frequency shift make no doubt for the observation, and that has finally even got into the science, saying that the alkaline Earth's main group atoms bind in octacovalent via d orbitals like a transition metal, calcium, strontium, and barium. Is it only for CO? Could it also be for other metals? What about N2, which is valence isoelectronic? According to the calculation, yes. The neutrals, triplet, the cations are here. For calcium and strontium, they have D for D symmetry. For barium, they have D for H symmetry. But all of them are stable with regard to loss 1 towards 1 of loss 0. And here is the vibrational spectrum just for the calcium. We got it for this. I skip a lot of data we got for calcium, strontium, barium for the neutral. Good agreement of the frequency shift. Look this for the isomers. Look this of this isomers with regard to the calculated one. In case of calcium and strontium, the calculated values agree also quite well. The experimental data for barium was difficult to interpret, therefore we don't show them. This finally got it into Nature Communications. This is the MO correlation diagram, very, very similar. The degenerate Egerado HOMO has 2 unpaired electrons. The A2 Ungerado, which is below, is a ligand-only orbital. So we actually have 16 electrons again. Very, very similar situation. And also the calculation showed that the binding again goes via the d orbital. This is a contribution of the pi-back donation, and this is the one of the sigma donation. The s and the p orbitals have little relevance. Let's go one step on. Are there other stable complexes besides the octa-coordinated species? What about sandwich complexes? You know that sandwich complexes play a special role in transition metal chemistry. Again, we first run the calculation for calcium, strontium, and barium, found yes. Now we should get systems which have 6, 12, 18, and 20 electrons, but we are not afraid of it anymore, having D3 symmetry for all 3 metals. They should be stable with loss of 1 CO or 3 VOO. Even after temperature and entropic correction, they should be stable. And this is an agreement. I just show the strontium spectrum. The whole paper, the whole work was published in Angewandte Chemie. Now we have an old diagram, a correlation diagram, where we did it in D3H. We found that D3H is only a little different from D3, but it makes the orbital symmetry, the correlation diagram, more simple. And now the HOMO is not degenerate, and therefore it's a singlet species, but we have 1 orbital, which is again ligand only bound, has 2 A2 prime symmetry. So although we have 20 electrons, only 18 electrons are available for the metal, and all the valence electrons of the metals are occupied. And if you make it MOCD, an EDA NOCV calculation, you see the main contribution is the back donation into the metal D, and even the next one are the D orbitals. The S and the P orbital have little relevance of calcium, strontium, and barium, mainly bind to the d orbitals. That was the time when I stumbled into this paper. It was published in 2006 by the experimental group of Sert Harder, together with the theoretical group of Mercedes Alonso. Stuart Harder is a Dutch chemist working in Erlangen in Germany. Mercedes Alonso is a Spanish chemist working in Brussels. We are all working in Europe now. So it's a European work. So I said, I contacted them because so far all the chemistry was done either in low temperature matrix isolation or in the gas phase. But this was now real chemistry. They got an X-ray structure of this complex and they published it with this kind of interaction, and I immediately recognized this could be an arene complex. And I contacted Stuart Harder and said, let's have a look on it. And we did some papers, and he was in the beginning quite reluctant and said, I don't believe it. Well, I think I want to make you a believer, and I showed that indeed the d orbitals are mainly involved in this bonding. We did some work, but then we did something which was really a sensation, and I immediately jumped to it. He showed us an X-ray structure where N2 is bonded to calcium. N2 activation is a very, very hot topic, and we did first a beta analysis of this paper, which this year got us into Science. And you see immediately that there is a nice bonding interaction between the N2 and a cyclic structure. But what are the orbitals of the calcium which are involved? You cannot see it from the EDA and CV, but you can get it from the EDA and CV. You cannot get it from the beta analysis. And you can see we run the calculation with two types of fragment, which you can do. It's very flat. You either look interaction of a double cation with a double anion or a neutral species. And in both cases, you find that the largest orbital interaction, in this case, we had a lot of different contribution, comes of an orbital where you have this calcium and where you have this calcium. And if you looked on this term, first for the anion, and you look that this is the interaction from the N to 2 minus into orbitals of the ligand, you immediately recognize that at the calcium, these are d orbitals. And if you do the same for the neutrals, it is the vice versa. And now you can see at the calcium, these are d orbitals which bind from here to here. And the associated charge flow comes from here, from the red to the blues, from the ligand to the metal, but it involves d orbitals of calcium. So we made a large paper last year on the valence orbital of the alkaline earth atom in all kinds of molecules. And I just point out a very simple system. Diatomic, calcium oxide, strontium oxide, and barium oxide. We compare it with beryllium and magnesium. And here's the hybridization of the pi and the sigma orbitals, which you get. In case of magnesium, you find only a sigma, in the NBO, only a sigma orbital. In the other case, you find double bond. If you look on the hybridization of the orbital, you find it's the S and the P, mainly the S and the P. Here it is the P. As soon as you go to calcium, it immediately jumps. The pi orbital is essentially D. Look at the ratio. And here it is the main contribution of the D, the same for strontium and so on. So even the NBO analysis tells you already there is a big jump between beryllium and magnesium on the one hand side, which are main group elements or SP block elements, but calcium, strontium, and barium bind to this. And you can plot it. You can see these are the metal atoms, and you can see this is mainly a small contribution of the SP of the, and this is a P. But here it is now calcium, and you see in the sigma orbital, you see immediately this is not a P. This is a d orbital. This is a d orbital. This is a d orbital, and it's for the sigma. And here for the pi, it is a dp pi orbital, a d orbital. So. And the latest work in this, which just came out a few weeks ago, we did metal CO bonding in mononuclear transition metal chemistry. We did group 4, 6, 8, We did all of them, including calcium. And in this paper, we showed why calcium, strontium, and barium are like a transition metal. As a neutral species, the electronic ground state has both electrons in the S. The first excited state of calcium and strontium is actually the P below the D, and barium is already the D. But in most cases, these metals lose an electron, bind, even a neutral molecule, bind like cations or dications. And as soon as you go to the cation, the third excited state is the D state. The electrons go to the D. The electrons go to the D. And here with little excitation, and it goes to the D. But the D are much more diffuse. And so for the overlap better. So therefore, the covalent bonding contribution comes from the d orbitals, which have a much better overlap with the orbital of the ligand. And this is what we propose as a new periodic system, that the d block shell should extend to calcium, strontium, barium, and most likely of radium. And we propose them, and we will see if people will accept it. This is the very latest scientific talk. It just came out in Angewandte Chemie, because we continue and going on the transition metal chemistry of calcium. And we could show that in the gas phase, calcium binds also in bimetallic cases, as is known for other transition metal with calcium, with iron, with a bridging CO that is published. We have it with other metal. And what we are working right now, it's not even submitted yet, is what happens when you have two alkaline earth metals, but you will find it in the literature in the near future. Summary and conclusion. Rules are rules, but there are no laws. So you can go beyond it, and you shoot a background. The alkaline earth elements, calcium, strontium, barium, bind through their D. They not only bind, they usually bind through their valence d orbitals, exhibiting the full scenario of genuine transition metal compounds. The n-electron rules of language, they remain very helpful, but they should be used in conjunction with the quantum theoretical analysis of the electronic structure. And when one counts the number of electrons that are engaged in the bond to an atom, one must consider the symmetry of the orbital. This holds also for the octet rules. PF5 follows the octet rules if you look on the orbitals of the ligands. And finally, the EDA NOCD method is a very powerful tool to analyze this and provides a quantitative estimate of the pairwise orbital intact, which is nicely visualized by the electron, by the calculated deformation densities. My acknowledgement go to my co-workers, and I am delocalized with an electron. These are the theoretical co-workers. This is my experimental co-workers, nicely one. These are the institution which gave us the money for the research. But the final word, of course, should go to Ashoka. Ashoka, this is how we see us now. I am retired. I retired already formally 7 years ago. Now you join me by retirement. But I tell you, it's not how old you grow, it's how you grow old. I wish you all the best for hopefully many, many years to come, and that we can share our life, the rest of our life in this way. Thank you very much for friendship and for your great science, and all the best. And so thank again for the invitation that I could give this lecture. Thank you.
I am sure that all of you will agree that this was a very interesting talk, and therefore let us record our appreciation of Professor Franking. And personally, I would like to express my sincere thanks to the organizers, Professor Ironan, Professor Debashish Das, and Professor Tilaga for this opportunity to chair this session, and I look forward to the remaining sessions. And with that, this session comes to an end. Thank you all.
Thank you, Professor Sebastian and Professor Krishnamurthy, for chairing the session. I suggest wherever you are, pick up your lunch and you can come back to Gather Town. We'll be there. Let's start the next session at 2. We have enough time now. So the Gather Town would be on now. So you can join in Gather Town. If you can write to Frankie, and any of you would like to make any comment about the talk, we can just for us.
If you have any questions, you can post it. And,
yeah,
we can get the answer for it.
Yeah. Okay, let's close this meeting. I will stop recording.